Hello and welcome to today's webinar. My name is Jennifer Caveney and I will be moderating today's presentation. Before we begin, just a few housekeeping notes. Following the presentation today, there will be some time for some Q&A, so please submit your questions in the questions box throughout the presentation. The recording of this presentation will also be sent out within the next 3 business days to all registrants. So we have a couple of excellent speakers here today from our Eurofins team. Our first speaker today will be Dr. Jamie Willems, who is the Manager of Special Analysis at Eurofins SFA Laboratories in New Berlin, Wisconsin. She received her PhD in food chemistry from the University of Saskatchewan and specializes in various gas and liquid chromatographic techniques and has experience with custom method transfers, method development, suitability studies, method validation, and flavor and fragrance analysis. Our second speaker will be Dr. Chris French, who is the Laboratory Director at Eurofins SF Analytical Laboratories, located in Tustin, California. He received his PhD in physical chemistry from the University of Virginia and specializes in spectroscopic techniques, including FTIR and energy dispersive X-ray spectroscopies, which he uses for failure analysis, for material identifications, and forensic investigations. Coming from a background in medical devices, Dr. French also served as study director for GLP compliant projects. So to kick off today's call, we want to start with a question for the audience. According to multiple industry sources, what was the #1 consumer complaint in food and beverages in 2021? Foreign bodies, unlisted ingredients or allergens, or damaged packaging? All right, we'll give it a few seconds here. All right. All right, so the answer is actually A, foreign bodies. Foreign bodies are the biggest single cause of consumer complaints received by many food and drink manufacturers, retailers, and enforcement authorities. The accidental inclusion of unwanted items may sometimes occur in even the best managed processes. Foreign bodies in foods are therefore a matter of concern to all food and beverage manufacturers and retailers. Thank you all for participating in today's poll, and now we are on to our presentation. Jamie, whenever you're ready.
All right, thank you for that introduction. So as mentioned, I'm Dr. Jamie Willems, and today, along with my colleague, Dr. Chris French, we are going to be presenting on analytical chemistry in support of forensic and investigational studies. So to begin with, I'm just going to give a very brief background on who we are and what we do, and then I'm going to outline kind of how we approach these non-routine investigations, and then give some examples of some of the different types of projects we tackle here. So Eurofins SF Analytical is a group of 5 labs under the Eurofins umbrella here in the United States. Today we're going to be focusing on kind of the investigational work that is done at SF Analytical here in New Berlin, Wisconsin, where I am, as well as our partner lab at SN Special Analysis West in Tustin, California. So Eurofins SF Analytical offers a variety of different testing, including some routine offerings such as environmental, food chemistry, monographs, feed, animal feed studies, but we also offer what we call specialty testing. This is our non-routine investigational analysis. And this can cover a wide variety of different studies. Basically anything that doesn't fall in kind of your routine standard testing buckets comes to our specialty teams. And so on this slide, I have some examples of some of the different kind of major groups of studies and projects we take on. So this can range from everything from foreign material characterization, product and packaging failure analysis, do a lot of off odors and off flavors, some clean in place chemical contamination, custom method development and client supplied methods, packaging migration, extractables and leachables. We also support and can support on legal products. our projects. And this gives just kind of a general overview of what we do. We also, a lot of the projects we do don't fall into kind of one of these neat buckets, or they might kind of bridge the gap between them. So how do we handle these kind of unique situations? Well, there's kind of a very general 4-step process. So when we're dealing with some of these non-routine, non-standard testing, the first thing we want to do is work directly with you to gather information about the issues you're seeing or the project you want to work on. That is, what is the actual goal of the testing? What do you need, or what do you want to see as an outcome? From there, once we've gathered the information we need, we can develop a very specific testing plan and a quote to specifically address that issue or that goal. From there, we execute the testing and then provide a summary report. This is not just like a typical C of A, but a full-on report with details about what testing was performed, summaries of the results, and interpretation where applicable. Additionally, we also offer interpretation of results, including direct communication with scientists where needed, to help kind of walk you through what we found and help you address your problem or solve the current issue you are facing. And so now I just want to give kind of a general overview of some of the different types of projects we take on with some examples, and then I'm going to hand it over to Dr. Chris to give some additional case studies. So as was seen in the poll, one of the big complaints that we see is foreign materials. And so what we offer is what we call foreign material characterization to help identify these objects. And so I'm sure you're all kind of generally familiar with what we mean by foreign materials, but broadly defined, foreign materials are objects or materials that is not intended to be present in a starting material, ingredient, formulation, or finished product. This can also be, for example, you might find something on the floor of your processing plant or in your office that you don't know what it is. You need to characterize it, say an unusual powder or a pill, maybe in your shipping warehouse on the outside of packaging. That also falls kind of under this umbrella. And broadly speaking, foreign materials can be defined in two main classes. The first of these is extraneous matter, and this is material that's definitely not supposed to be present in a product. So for example, plastic, glass, metal, plant material. Some other examples include things like bugs. We see those on occasion, bug eggs. In fact, one time we even had one where someone found a tooth in a product. So basically things we know are not supposed to be present. The other kind of broad category is product related. Technically things that are supposed to be present, but maybe in a form that is not desired or unideal. For example, conglomeration of ingredients or an inhomogeneous product can lead to clumping and weird pockets of color or texture or oil that is unappealing and may appear foreign. Or another example of this would be degradation of raw materials, resulting in odd textures or colors. So as an example of one of these foreign material characterizations, on the slide, I have pictures of the inside of a can. And what happened here is the customer was producing a canned meat product, and it was canned in a brine. And what they were finding over time and getting complaints about was bubbling or the formation of blisters in the can lining. So you can see here we have a high magnification image of some of those blisters. You can see that's not really an appealing look. And so we worked to try and figure out, okay, what was causing this blistering? And so what we did is we took these high magnification images, we opened up the blisters, and what we found inside was this crystalline-like material. And then what we took that material and we analyzed it for its elemental composition using a laser-based elemental analyzer where we could target very specific locations. And we found that this material was primarily comprised of organic components, carbon, oxygen, hydrogen, but it also contained a significant amount of sodium, And we were also able to isolate some of this material and analyze it by FTIR. And what we found was these crystals appeared to be the brine that had solidified. And so when we took a closer look at these cans, especially the ones with the failures, what we found was that the lining was not sealing properly to the can around the lip of the can. And so what was happening is as these were being stored or transported, some of that brine was leaking between the can wall material and the lining. And then the brine was getting in there and then forming these crystal deposits, resulting in these blisters. And so based on this, the client could go back, fix this problem, improve their cans, and prevent this from reoccurring. So this was kind of like a combination of a foreign material characterization and a packaging failure analysis. And this was a case where the foreign material was technically product related, it was the brine, but it was in a place, in a form where it shouldn't be. And so when we do these foreign material characterizations, we use complementary techniques. And we can target these techniques and select them based on the specific problem or project we are addressing. So as I showed on the previous slide, there we use multiple techniques to ID not only the foreign material, but the issue. And we, like I said, can target this to the specific issue. So some examples of these include high-resolution microscopy, laser-based elemental analyzer for bulk and targeted elemental analysis, FTIR analysis for the evaluation of what organic functional groups and by extension organic compounds are present, ICP, OES, or MS for low-level metal analysis, and so on. So we can choose specifically for what we need. So another example of this on the slide, I have some gummies. And these gummies were part of a pilot production. And what they found was they were seeing all these little specks throughout the gummies. And these were very small specks. They were very hard to see with the naked eye. So you can see the gummy in the middle here. If you squint, there's a little dark speck right on that gummy. And that was one of the bigger specks they were observing. But they really needed to know where these specks were coming from. So what we did is we took these gummies and we did some high magnification microscopy, and we were able to get good images of the particles. So I have an image of one of the particles here. You can see its shape and color. And then from there, we were able to isolate them and analyze them by FTIR, which identified these particles as PTFE. We were also able to confirm their identity using the laser-based elemental analyzer, which confirmed the presence of carbon and fluorine. And so using this approach, we were able to identify the particles and we were able to analyze multiples to confirm they were all coming from the same source. Another major group of investigative projects we run are the off odors and off flavor analysis. So aroma and taste are really important parts of consumer appeal of food and related products. And off odors and off flavors are not only off-putting to consumers, but they may be a sign of a serious problem with a product. And there's a lot of different potential causes of off odors and off flavors. But some of the common ones we see include packaging issues. For example, putting a product in incompatible packaging can result in leaching of components from the packaging material into the product. It can also result in what we call flavor scalping, where the packaging material actually absorbs flavor components from the product, resulting in a change in the flavor profile or a reduction of flavor compounds. Another common source is ingredient issues, maybe a bad ingredient resulting in an off flavor, or perhaps a supplier has changed formulation, resulting in just a change in the overall profile. Chemical or microbial contamination can lead to rancid or chemical or solvent-like off flavors, as well as issues during transportation and storage. For example, heat abuse, temperature abuse can accelerate spoilage and rancidity. And a lot of these issues can kind of overlap with each other. And so just an example of how we approach these types of studies. So when we're looking at off odor analysis, we're looking at the volatile profile. That is, what chemical components of your product can make it into the air, reach the nose, reach your palate, and create that overall sensory experience. And so what we do when we're doing our off odor and off flavor analysis is we compare the volatile profile of different products. And we do this using an approach called gas chromatography coupled with mass spectrometry, or GCMS for short. And I'm not going to go into a lot of detail here, but on a high level, this works by collecting and concentrating those volatile components, separating out these different chemical compounds, and then detecting them using mass spectrometry. We can also use mass spectrometry to help identify these compounds as they produce very distinct mass spectra, which we can compare to our library to get information on the identification. So as an example, typically what we're wanting to do here is compare a good sample to the bad sample, so a control that is not exhibiting the off odor or off flavor. And in this way, we can look for differences between the control and the suspect that could be causing your off flavor. So for example, in this slide, what we have here in the bottom is what we call chromatograms. These are the profiles of the different volatile compounds present, and each of these peaks represents a different compound. And so we had one client who was making a batch of cookies, and they noticed in their sensory testing that they had a fatty or rancid aroma and flavor. And so what we did is we evaluated the profile of a control sample, the same type of cookie, but from a different batch that didn't have the off note, as compared to the off flavor sample. And what we found is that the off flavor sample contained a number of compounds not present in the control. So on this chromatogram, on the bottom in black, we have the control sample, and on the top in red, we have the suspect sample. And as you can see, there are three very distinct peaks there not present in the control. And these were identified as caprylic, capric, and lauric acid, which are free fatty acids. And these compounds are known to be associated with rancid fatty aromas. As well, taken together, these three fatty acids are the major components in coconut oil and coconut-related products. And so using this information, they could trace the issue back to a rancid coconut ingredient. And so in this way, we were able to help identify what was going on with this product. And so what we're doing here is we're looking for the differences in these profiles. So in this case, we saw the presence of additional peaks resulted from rancidity, but the reverse could be true as well. For example, another cause of an off flavor, maybe during production, an ingredient was missed, a flavoring was missed, such as vanillin. In that case, we could see, oh, the suspect sample is missing vanillin when it is present in the control. Somewhat related to off odor and off flavor analysis is complete clean in place contamination analysis. And so clean in place chemicals are a very key component in food safety and beverage production, ensuring that lines are clean, that we don't have cross-contamination or microbial contamination. And so they are critical in food and related production. However, in some cases, insufficient rinsing after cleaning or other situations can lead to the contamination of products, resulting in quality and or safety concerns. And so in these cases, we can do a more targeted testing, looking for the presence of specific cleaners through various approaches tailored based on the specific cleaner composition or safety data sheet. So for example, a lot of these cleaners are acids or bases. For example, sodium hydroxide is a common cleaner. Phosphate buffers are often seen in these cleaners. And in this case, what we can do is we can compare a sample suspected to be contaminated versus a control sample known not to be contaminated and evaluate them for things like pH, acidity, alkalinity, anion screening, for example, the presence of phosphate anions, metal analysis, and look to see if there are differences, elevated or lower levels of certain of these compounds in a potentially contaminated sample. Additionally, though, we can also do targeted chromatographic analysis, looking for cleaners that perhaps, for example, have surfactants or other ingredients that we can specifically target and look for lower levels of. So, for example, we had a client had a chocolate protein product that they suspected might have been contaminated with a surfactant-containing cleaner. And so what we did here is we obtained a sample of both a control product that they knew was good and a suspect sample that they thought was contaminated, as well as the cleaner itself. And using the cleaner, we were able to analyze it by LC-MS and find what we call a marker compound. And this compound we could use as an indicator for the presence or absence of the cleaner. And from there, we developed an LC-MS method to look for lower levels of this particular marker, and by proxy, the cleaner in question. And unfortunately, in this case, we did detect the cleaner in the finished product, in the suspect finished product in ppm levels. And so they did have an incident they needed to address. Another example of a clean-in-place contamination analysis, this one kind of overlaps a bit with our off-odor and off-flavor analysis, showing kind of how these projects can be flexible as needed. But in brief, a client, as part of their cleaning process, they were scrubbing something with a sponge, and then they did their clean-in-place process. Unfortunately, after doing that, they determined that a small piece of sponge had gotten lodged in a nozzle used for sample production, and that nozzle sprayed gas into their product. Unfortunately, the sponge piece was no longer available, and they wanted to confirm whether or not they should be concerned about potential contamination of the product. And so what we did, as the original piece was no longer available, we obtained a fresh sponge of the same sponge that they were using, and we subjected that to a laboratory scale version of the cleaning process. The same chemicals, times, and temperatures as close as we could mimic in a laboratory scale in order to see the impact on the sponge. So for example, on the bottom of the slide, I have the original green sponge prior to treatment, and then the black is the sponge after all that of the laboratory scale cleaning process. As you can see, it's become quite charred from the presence of the chemicals and the heat. And what we did is we took the untreated sponge, the treated sponge, and then control and potentially suspect samples, and we analyzed them by GC-MS to look for the presence of any volatile compounds that might be contaminating the sample. Unfortunately, in this case, we didn't see any evidence of contamination of the product from the presence of the sponge, and we're able to give them some peace of mind. And so in addition to those previous examples, we also do custom method specialty chromatography development. So like I talked about with the clean in place, with the surfactant-based cleaner, where we developed a method to look for that specific cleaner, we can do that in other products as well. So for example, you might need this, say you're developing a new product that you want to bring to market, and there's currently no available release testing routinely available. So this might be, for example, a new or novel analyte or matrix. Maybe you're developing a gummy with a specific plant extract, and you want to be able to measure the active ingredient or the active compound so you can ensure the quality of the product. And so we can help with that through the development of custom chromatography methods. This can be full method development from scratch. If you don't even have a starting point, we can help with that. Or you may have an in-house method that you're already running, but now you just need it validated and run at an external lab. We can also help with method transfers. So we can do full method development, method validation and verification, and after that, we can even offer routine testing for batch release as needed. And as a companion to chromatography, we also offer unknown analysis using mass spectrometry. And this allows for high accuracy mass determination of compounds. And why this is useful is we can use it for unknown contaminant analysis, ID confirmations, and we also offer custom extractable and leachable studies. And in tandem with this, we can use MSMS to allow for further structural elucidation through the use of fragmentation patterns. So if you're not familiar, mass spectrometers work by ionizing compounds and then separating them based on their mass-to-charge ratios. And using the mass-to-charge ratio, we can determine the molecular weight of a compound. And very broadly speaking, we can classify mass spectrometers into low resolution and high resolution, where the difference is how accurate and specific we can get on the mass-to-charge ratio. For low resolution mass spectrometers, you're typically looking at nominal or mass to the nearest whole number, while high resolution mass spectrometers can get you much more and more additional decimal points. And why is that beneficial? Well, this can allow for greater structural elucidation and differentiation between compounds, even with the same nominal mass. So just as an example, here on the slide, I have two very common compounds that are found in a lot of food products, and that's caffeic acid, a phenolic, and glucose, a carbohydrate. And they both have the same nominal mass of 180. However, if you extend that value to further decimal points, you can see they actually differ by about 0.02 grams per mole. And therefore, we could differentiate these two masses using high-resolution mass spectrometry. So an example of how this is used, in this case, we had a client who was doing an in-house stability study using their product, a new drug substance, in a newly developed packaging material. And what they found over time was two unidentified peaks appearing in their chromatogram during the analysis. These peaks didn't line up with their analyte of interest or any of the known degradants. And they were doing all this work in-house, and they didn't know, and they didn't have a way to identify these peaks, and really needed to know, okay, what are they, where are they coming from, what's going on? And so that's where we stepped in to help. In this case, we took their method and converted it to be MS-friendly, as the method they were running was not compatible with mass spectrometry. So we adjusted it so we could run it using mass spectroscopy, but keeping the same general separation and elution so we could still identify the peaks they were interested in. And when we did this, we ran it by high res, and we were able to identify those two peaks as caprolactam and 1,4,7-trioxyacyl tridecane 813 dione. And these two compounds are both common in plastics as plasticizers and are known to be leachable from plastic materials. And so from this, we were able to determine that it wasn't a degradation of their pharmaceutical or analyte, but rather an issue with leaching from their packaging. And using this information, we were able to obtain reference standards and quantify exactly how much of each of these contaminants were present in each of their samples, and therefore they could use this information to further plan new stability studies and adjust their packaging as needed. So now I'm going to hand it back to Jen before we hand back over to Chris.
Great, super. Thanks, Jamie. Great information presented so far. So just, question just as we're working through different technologies and different pieces. So if contamination, if a client is suspecting that contamination, their product is contaminated, what information should be gathered in order to expedite an investigation? Because oftentimes I would assume, those are pretty urgent.
Right. So yeah, I can take that question. From a high level, the more info you can get us, the better. It can help us plan and target the investigation. But from a more general answer, it really helps if you have an idea of what the suspected contaminant is. If you have, information, let's say Perhaps it's a cleaner. If you have, for example, an SDS or a C of A, information on the suspected amount of contamination is helpful, or a desired limit of detection, as well as product information. So what product was contaminated, how much of that product was contaminated, can also help us plan.
All right, awesome. Thank you so much.
So now I'm going to hand it over to my colleague, Dr. Chris French, to give you a few, talk a bit more about SN Special Analysis West.
All right, thanks, Jamie. Again, my name is Chris French. I'm technical director here for Eurofins Special Analysis West. I'm really excited to be part of this fairly small team, and the reason is a lot of the analytical labs out there may have to process 300 samples, get the reports out the door, but we're a niche lab. We're very different in the way that we will engage a customer face-to-face, sit down with them a lot of times in the conference room, understand their problem at a deep level, and then go into the laboratory and do a deep dive and figure out exactly all the complexities, potential root causes, et cetera, et cetera. And, you know, as a chemist, you put me in a room with a couple $1 million of state-of-the-art equipment, let's face it, it's just plain fun. But where we came from, we've actually been around for about 40 years. SNN Labs was created by Dr. Spingarn, who realized that there's kind of a unique opportunity because, you know, what do you do when you have a 20 micron particle and you have no idea what it is and where it came from? Well, in '83, there just weren't any labs that would do that. So, you know, he brought out an electron microscope, infrared microscope, and since then, you know, basically word of mouth starting here in Orange County, California, and we've grown to a client base of thousands who really appreciate the level of expertise that we offer in terms of complex, you know, chemical contamination, et cetera, problems, and they can't be solved anywhere else. Next slide. I'm not going to go into this a lot, but there's a lot of overlay between the equipment we use here and they do in Jamie's lab in New Berlin. Infrared spectroscopy is good for molecular fingerprinting. GC is great for volatiles. Electron microscope we have for high resolution or high magnification imaging also gives you a really nice elemental profile for what you've got. And then a really expensive optical microscope that can give you full color imaging to up to 2000x. Next slide. Basically, I'm going to run through a couple of case studies just to show the types of things that we work on and how we approach them. We want to leave some time at the end for questions, so I'm going to go through these at a pretty good clip. But if you have any questions, you know, please feel free to use the chat and we will follow up as we can. To Jamie's point, one of the biggest problems facing multiple market sectors is what is this material on my product, or what is this material in my product? And I don't care what market sector you're in, these issues pop up invariably. So we've got medical devices like, oh, I've got an intraocular lens. What is this black particle in my lens? Or an aerospace client might come in and say, oh, I've got these fines building up in our coolant oil. Or someone might bring me a cherry pie and say, why is the crust turning black on top? What the heck? You know, especially allegations of like animal parts, etc., or mold, right? So this case study one, it looks like this chicken jerky has just gone moldy, right? What the heck? It's going through the same process of, you know, as the top control article here, but it looks like it's just completely molded over, but really couldn't be further from the case. So we take this material, take a little bit of that white material off, and we use infrared spectroscopy and EDX spectroscopy. And you can see in the upper right there is an infrared match showing the white material is primarily creatine. You know, that's an amino acid. It's like, what the heck? And then other parts of the white material are like magnesium phosphate. In the lower right there, you can see an example of an EDX spectrum showing the sort of elemental composition of that material. But then, okay, so it's not mold. What is it? So one of the things we do for clients is we go into the literature and say, okay, so we got this basically a meat product. Why would we be seeing creatine and phosphates? And it turns out there's a vast literature saying, wow, this happens all the time. And why is that? And how do we stop it? It turns out, again, we can make some good solid recommendations based on our deep diving of the literature and really help the client to prevent these problems moving forward. Next slide. Another thing that I wasn't aware of as a chemist is the curious occasions where rogue architectural materials will run around at night and smash into your car. I was completely unaware that this happened, but a lot of times, you know, someone will file an insurance claim for 10, 15,000 bucks, and the adjuster will be like, this doesn't look quite right. You know, that gash is a little bit too high for a hit and run. I know you say you're parked outside your apartment all night, but we're gonna have a look. And as it turns out, you know, the accident investigators will look at the position, the potential velocities involved, and then they'll bring us a paint transfer sample. And it turns out a lot of people don't know that architectural paint looks very different in terms of chemistry than an automotive paint. So if you claim that you're struck by an automobile, right, bring in the paint here. We use infrared and EDX spectroscopies, and we're like, look, this is an alkyd. Most top coats are acrylic urethanes. We don't believe this is consistent with hit and run from an automobile. Especially with, if you look at the bottom right, the elemental analysis of that paint transfer, lots of mineral fillers. There's a shifting trend now toward, you know, matte types of paint, but historically people want shiny cars, right? So if you got a lot of mineral fills, the paint's not going to be shiny. So ultimately, we think that this particular example was one of those road posts that go running around neighborhoods at night. Next slide. In this particular case, we had a blast furnace operator, had some fairly suspicious syringes on his desk. So, you know, the manager bring these into us. We do the chemical analysis using infrared, and you know, it's brown, so it doesn't look like a normal drug, but clearly by the infrared spectrum there on the lower right, it's meth. And then do the deconvolution analysis, and the second component is blood. So I was like, why is there blood in meth? You know, I've never used meth, so I don't know what the industry best practices are, but apparently, according to one of my police officer friends, and users of meth sometimes will pull a little bit of blood to make sure they're in the right place, etc. And that explains that mixture. Next slide. Other types of things we do are material failure analysis. This was a sample from a large fast food chain, and they were experiencing rapid degradation of their fry bin supports. So we clipped a little bit off. Looks like things were just decomposing for no apparent reason. And but looking under the microscope, it looked like a lot environmental stress cracking was going on. What we did was we built a Bergen jig, which was basically taking some polymer coupons, straining them over those aluminum posts you see there on the lower right, and then wrapping them with the chemicals that are in that environment. So like there are wiper materials they use to wipe down the fryer, isopropanol, ethanol for the disinfectant, canola oil, etc. And what we're able to do is, if you see that picture there with all the lines going horizontal, That's environmental stress cracking. We were able to positively isolate which of those chemicals were not compatible, so they were able to avoid that in the future. Next slide. Another thing we do is offer a lot of litigation support. So if you have an attorney, need some scientific background to support your data, we do depositions, court appearances, written statements, etc. In this particular case, a woman was walking through a grocery store, claimed she slipped on some kind of liquid in the aisle and needed some kind of science to back that up. So the attorney brought us in the shoes, took a little bit of what appeared to be a stain there on the sole, and we found that it was contaminated with a natural oil, and they were able to take that information and support their claims in court. Next slide. The other suspected food contamination that happens with surprising frequency are debris that you find in a commercial water bottle. You wouldn't think that would happen often, but we work with some of the largest commercial bottlers out there. You get something back that looks like this pinkish worm-like thing that the client found in their water bottle, and they're like, where did this come from? So they sent us the water bottle with a worm-like creature swimming in the bottom. We pulled it out, isolated it, used infrared and mass spectroscopy to identify what this material actually was a proton pump inhibitor. And shockingly, the Prilosec that was in this pill happened to be used by the client at the same time. So a lot of times people take a swig of water, take their pill, and it backwashes into the bottle. The bottle gets returned to the commercial bottler with complaint, and they have to decide what to do with it. And we give the client clear evidence that this was not on them, but rather the person trying to swallow the pill and let it slip back into the water. Next slide. Suspected contamination of food is, again, one of the things that we do pretty well here. In this particular case, we got a huge shipment of grapes coming up from South America, and they think they left the cargo door open, but they're not sure, and they wanted to figure out whether or not they could still sell this 1 million pounds of grapes. And so they sent us the grapes. We used a solid phase micro extraction technique that has phenomenal sensitivity. We're talking parts per billion, possibly even lower than that. Overlaid it with reference diesel, and you can see from that fingerprint overlay in the upper right, clearly there's diesel exhaust on both batches of the grape, and they unfortunately had to scrap that entire shipment. All right, next slide. So last case study, and then we'll have some time for questions. So we're here in Tustin, California, and we have a local bread market in Santa Ana, just up the road from us. It was selling Christmas bread one day, obviously near Christmas, and people who were eating the bread were immediately getting sick. Some of them were going to the hospital, some were having hallucinations, and obviously handed it off to the crime lab, and we got a part of that, and we extracted that into some methanol and used liquid chromatography to identify that the offending drug was actually a synthetic cannabinoid. So in this weird particular case, someone had actually laced the bread at this bread market in Santa Ana with JWH122, the cannabinoid. And so we were able to, 2 weeks faster than the crime lab, tell the public exactly what had happened in that particular case. So again, appreciate the time. We're going to have some time for questions here. Wrapping up a little bit early, and Jamie and etc. will take the lead from here.
All right, perfect. Thank you so much, Chris. So many different case studies, so many different different things reviewed, so thank you. All right, so first question we have from the chat, what is the best analytical approach to determining if a contamination occurred?
Yeah, I can grab that one. I guess the short answer is it depends. Kind of as we discussed earlier, the more information you can get us, the more targeted we can be in our analysis. So for example, if you suspect a contamination with a cleaner that contains a surfactant, we could do a targeted LCMS method. But if you suspect it was contaminated with something, for example, that has a high metals content, we could do elemental metals analysis, or similar to what Chris just talked about with the bread, where they found the cannabinoids. So the more information we can have on the front end of what you suspect or what information you have about what's going on, the more targeted we can be.
Okay, perfect. Thank you. Another question from the chat. How long does a typical beverage investigation take?
Really depends on what type of beverage investigation, right? A lot of times we'll have packaging failure analysis, which can only take a few hours to figure out why their packaging or their beverage is leaking. Off flavor, off odors takes a couple days. Jamie gave some examples of that. Using chromatography, we essentially extract odor and flavor causing molecules and then run them through a gas chromatograph and see what's different. And obviously, if you've got foreign materials in there, it only takes a couple hours to pull that out. Rinse them up, clean them up, and then do things like infrared spectroscopy, EDX spectroscopy to identify what it is. But yeah, so anywhere from about 2 hours to 2 months for, let's say, a stability study. That's a good question.
All right, thank you. All right, one other question. Have you come across investigations where the exact culprit could not be identified using existing reference libraries? Do you make educated guesses or attempt structure elucidation?
Yeah, I can grab that one as well. So yes, we have seen this, and it kind of depends. All these projects are pretty different, but it kind of depends on the situation. But with the information we have, we can often give additional details. So for example, if we're running something by FTIR, we might be able to give information on what functional groups are present, say, for example, amide bonding, carbonyl groups. But one example we had of this actually with high resolution mass spectrometry, similar to the contaminant case study I showed where we found the surfactants or the plasticizers, is a very similar situation where a customer was analyzing a novel pharmaceutical drug that they were developing, and they were finding similar unknown contaminants or unknown peaks in their analysis. And because there was no commercially available reference material or anything like that available for this compound because it was new and novel, we weren't able to get full structural elucidation, but we were able to determine that these contaminants, these unknown peaks, shared structural features with their analyte of interest, and therefore were most likely degradants of that analyte and not from an external source, such as packaging leaching.
Yeah, we get a couple of things that stump us here too. A lot of times people will bring us a material and it's like clearly a microorganism, but we have no idea which one. So we have partner labs in microbiology who sort that out for us. And then there's a lot of new engineering polymers out there that just simply aren't on our radar. But a lot of times what the company might do is bring us 6, 8 different polymers and say, well, which one matches? Even though we can't quite figure out what the polymer is because it's probably brand new to the industry, we can give them fingerprint matches and say, okay, we don't entirely know what this is, but it matches sample B.
All right, great. Thank you so much. Well, thank you both, Jamie and Chris, for today's presentation, and thanks to everyone who attended. This now concludes today's presentation. Have a great day.